this video, we'll show how genetic information is transmitted across generations. The information needed for the survival and proliferation of all living organisms must be passed from one generation to the next. We can take an example of a growing bacterial cell. These bacterial cells will divide by a process called binary fission. During binary fission, cells elongate and then separate to produce two identical resulting bacteria. Both the original and resulting cells have identical morphology, identical structural organization, identical biochemical activity, and identical requirements for survival and proliferation. The simple distribution of an existing set of essential proteins from a single parent cell between two resulting cells is not viable, because without new synthesis, the concentration of proteins in the cell would decrease by 50% each time the cell divides. This would result in cell death. Under optimal conditions for growth, the generation time for bacteria is 20 minutes. This means that a dramatic decrease of the protein concentration would occur within a few hours, and cell death would quickly follow. Therefore, bacteria must contain the information that is needed to synthesize and replenish their stock of essential proteins, and this information must be transmitted across generations. Let's look at the biochemical adaptations of bacteria to changes of carbon sources as an example of the synthesis of new proteins in the cell. We'll start with bacteria in the first phase of growth, where glucose is the only source of carbon available. Glucose in the growth media is taken up by the bacteria and broken down by glycolytic enzymes to produce ATP, the form of energy that bacteria use to survive. During this phase, the concentration of glycolytic enzymes is high and remains constant. However, concentrations of different enzymes that are not directly involved in glycolysis, for example, beta-galactosidase, denoted with the Greek uppercase letter, cannot be detected inside the bacteria. During the second phase of growth, there is a change in carbon source, and lactose replaces glucose as the only source of carbon available. The lactose is taken up by the bacteria and cleaved into a molecule of glucose and a molecule of galactose by beta-galactosidase, and the glycolytic enzymes that are still at high concentration will break down glucose and galactose to produce ATP. During this phase, we observe a quick increase in the synthesis of beta-galactosidase. The concentration of beta-galactosidase will remain high as long as lactose is the only source of glucose. During the third phase of growth, where lactose is replaced by glucose as the only source of carbon available for the bacteria, the concentration of glycolytic enzymes remain high and constant, and there is a relatively fast decrease in the concentration of beta-galactosidase as it is no longer needed by the bacteria. This experiment shows us two things. The synthesis of some proteins is dependent on functional needs, and our observations cannot be explained by a simple model where proteins serve as a model for their own synthesis. So now we need to ask another question. Is there physical evidence that hereditary information is transmitted through proteins? Proteins are essential for the functioning of cells, and each protein is characterized by three things: its amino acid composition, the sequence of amino acids along the polypeptide chain, which we call the primary structure of proteins, and the three-dimensional structure of the protein, which is determined by the primary structure. So, how are these proteins made? Previously, we observed a scenario where bacterial cells shifted from utilizing one carbon source to another, and it seemed like the protein synthesis machinery adapted quickly to produce enzymes necessary for utilizing the new carbon source. Given this observation, you might wonder, why is it unlikely that proteins themselves carry the information needed for the synthesis of other proteins? If proteins were to serve as a model for their own synthesis, it would mean that enzymes, which are proteins themselves, are needed to catalyze the synthesis of new proteins. However, this creates a logical problem known as the chicken and the egg dilemma. To synthesize a protein, you need enzymes, but those enzymes themselves are proteins that need to be synthesized. This creates a circular dependency. You need proteins to make proteins, but you can't make proteins without proteins already being present. In essence, this scenario would require an infinite supply of enzymes to initiate the synthesis of proteins, which is not practically possible. The information-bearing molecules we are looking for must encode the information needed to order amino acids in a specific sequence during protein synthesis. They must be capable of synthesizing new copies of themselves to transfer the information across generations. These molecules should be divided into units, with each unit carrying the information necessary to synthesize a protein. We now know that DNA serves as the information-bearing molecule, and within DNA, these units are called genes.